Hello friends, welcome to Live Biochemistry. I am Dr. Ashish. Today we will discuss about the fatty acid synthesis. This fatty acid synthesis, it is taking place in the cytoplasm of the cell. And here mainly the 16 carbon palmitic acid is synthesized. Now what is the requirement for the fatty acid synthesis? The precursor of fatty acid synthesis is mainly the 2-carbon acetyl CoA molecules and NADPH. Now what are the sources of NADPH? The main source is HMP shunt pathway, hexose monophosphate shunt pathway, which produce NADPH. So one molecule of glucose 6-phosphate will give 2 molecules of NADPH, which is required for the fatty acid synthesis. Another is isochronic isocitrate dehydratase that will convert isocitrate to alpha-ketoglutarate, and NADP is converted to NADPH. And third is Melic enzyme that will convert malate to pyruvate, and NADP is converted to NADPH. So the main is the shunt pathway. Minor source of NADPH is cytochronic isocitrate dehydrogenase. There is mitochondrial isocitrate dehydrogenase that takes part in the TCA cycle. Now, how fatty acid synthesis take place? Acetyl CoA, which is produced in the mitochondria, it is impermeable to inner mitochondrial membrane. So it will not cross the inner mitochondrial membrane. So what will happen? This acetyl CoA bind with the oxaloacetate to form citrate, and with the help this citrate can transport the inner mitochondrial membrane. So this is mitochondria. So acetyl CoA plus oxaloacetate will form citrate. Citrate will cross the inner mitochondrial membrane. It will come out in the cytoplasm as fatty acid synthesis enzymes are present in the cytoplasm. Then it is breaking down again to acetyl CoA and oxaloacetate. Now the acetyl CoA, which is come out in the cytoplasm, it is converted to 3-carbon malonyl-CoA by the acetyl CoA carboxylase enzyme, and it requires ATP, biotin and carbon dioxide. Here is the one reaction where biotin is required, and it is the key enzyme of fatty acid synthesis. So acetyl CoA carboxylase is a key enzyme of fatty acid synthesis. Like we have seen in glycolysis, PLK is the main enzyme. In beta oxidation, there is a cat1 enzyme. Carnitine shuttle transport is the main enzyme. Same way, in fatty acid synthesis, acetyl CoA carboxylase is the key enzyme of the fatty acid synthesis. Now, the enzyme for the fatty acid, it is a multi-enzyme complex. It is made up of 7 units of each monomer, and the functional unit is dimer. And so here we can say like this. Here there is a enzyme called as the ketoacyl synthase Another is Acetyl Tranacylase Malonyl. This is malonyl transacylase. So these three are the first required for the fatty acid synthesis, and this ketoacyl synthesis is attached with the cysteine, so it contains the sulfite group. While other units enzymes, so this is one monomer which consists of the 7 enzymes. One is ketoacyl synthase, acetyl tranacylase, malonyl tranacylase, then this fourth is dehydratase, then we have a enoyl reductase, ketoacyl reductase, then we have a ACP portion, acyl carrier protein, which bound with the pentogenic acid derivative phosphopentokene, and is a thiol group, so there is the acid group, and 7 enzyme is thioesterase. So we will see in detail how this works. And again there is a like DNA antiparallel, here also there is a 7 carbon units. So one monomer unit, so there are 2 monomer units in fatty acid complex, and the functional unit is dimer. So we will see that here. So from, suppose this is the head and this is the tail. So towards the tail side, like here, there is a ketoacyl synthase enzyme, ketoacyl synthase, then acetyl tranacylase, here there is a malonyl tranacylase, and here with the ketoacyl synthase cysteine group we bound with the acid. So this will react with acid group of the phosphopentokene. Here also there is a dehydratase enzyme, here there is a enoyl reductase, here there is a ketoenoyl reductase, and here there is a, this is ACP portion which contains the phosphopentokene which bind with this, and here it is the thioesterase. So the functional unit will contain enzyme like this portion only. This is the dimer. So if we break down this into 2, then this is 1 unit, this is a 1 unit which contains ketoacyl synthase, acetyl tranacylase, malonyl tranacylase, dehydratase, enoyl reductase, ketoacyl reductase, and thioesterase. Okay, so if we see, if we divide this into the 1 unit, that is a dimer, so if we take only this unit, how will it return, we will see. Okay, so it is like so here there is a thioesterase, this is ACP portion which contains phosphopentokene with acid group. Here there is a ketoacyl reductase, this is enoyl reductase, and this is dehydratase, and this portion it contains ketoacyl synthase, acetyl tranacylase, and malonyl tranacylase. Ketoacyl contains cysteine acid, and this is about the functional unit of fatty acid synthesis. Now what will happen? Then we have said acetyl is converted to malonyl-CoA. Now this acetyl CoA bind with the enzyme called Acetyl Tranacylase, and it will form the acetyl enzyme plus CoA effect. So what will happen here to this complex? So we can say that suppose this is a here first acetyl CoA first bind with the ketoacyl. This part acetyl CoA will bind with the this portion, and it is a temporary holder. Then this malonyl-CoA with the help of Acetyl Tranacylase, it will bind with the ACP portion here, malonyl-CoA, and it is from the malonyl ACP plus CoA SH. Now this acetyl CoA will bind with this malonyl-CoA. So here acetyl CoA plus malonyl-CoA will combine together, and with the help of enzyme ketoacyl synthase, that is the third enzyme. So first one acetyl carboxylase, then malonyl-carboxylase, now ketoacyl synthase. So 2 carbon acetyl CoA plus 3 carbon malonyl-CoA, they will combine here and they will form ketoacyl ACP, and here CO2 is removed. So now this ketoacyl ACP is formed. We will also take together how this beta oxidation takes place. So it is easy to remember. What happens in beta oxidation? This acetyl CoA first undergo oxidation to form enoyl-CoA, then enoyl-CoA with the help of H2O, it is converted to hydroxyacyl CoA. So in enoyl-CoA there is a 2 double bond, and in hydroxy there is a OH group was there. Then again it undergo oxidation. Here FAD was converted to FADH2, and here in hydroxy and with the help of NAD it is oxidized, and it will form ketoacyl CoA, which is broken down to acyl CoA, which is less than 2 carbon, and 1 acetyl CoA. So here exactly reverse. So first ketoacyl ACP will form with the help of acetyl CoA and malonyl-CoA with the help of ketoacyl synthase enzyme. Now this ketoacyl, what happened here? It is converted to hydroxyacyl CoA. Here with the help of NADPH, it is generated mainly in the assimilation pathway. It is converted to NADP, and ketoacyl CoA is converted to beta hydroxyacyl ACP. Now in this step, in beta oxidation, what was happening? This hydroxy was converted to enoyl. So to convert enoyl was converted to hydroxy in beta oxidation. Here reverse occurred. So there is a dehydratase enzyme is required. Dehydratase, and it will be converted to enoyl-CoA. Now in beta oxidation, enoyl-CoA was converted to acyl CoA was converted to enoyl-CoA. So again this enoyl-CoA will be reduced with the help of enoyl reductase. Yes, enzyme was ketoacyl reductase. So with the help of enoyl reductase, it will be converted to butyryl CoA ACP. Here also beta hydroxyapatite ACP, enoyl ACP, and here butyryl ACP, which is the 4 carbon atom. This butyryl ACP is expelled by the thioesterase. So this butyryl group is released. Now again what will happen? Again this butyryl, here there is a butyryl ACP, here there is a malonyl ACP. It will be condensed by the ketoacyl synthase. So every time 2 carbons are added. So after 4 carbon and 6 carbon, 8 carbon, 10, ultimately 16 carbon fatty acid complex, that is palmitic acid, is synthesized. So like again I revise. So in summary, we can say this is the one domain and this is another domain. So acetyl CoA bind with this ACP region, then it will be transferred temporarily to this cysteine site. So it is called by the acetyl tranacylase, so it is called acetyl plus enzyme will hold here, and this vacuum site now, so it is transferred. So here there is the FPP part, this vacuum site malonyl-CoA will bind, so it is ACP and malonyl-CoA will bind here and this side. So it is called the malonyl ACP, and here this is the acetyl group. Now these two with the help of ketoacyl synthase acetyl CoA will be transferred, and here at the ACP region it will form ketoacyl ACP with the ketoacyl synthase enzyme. Then it will be reduced to hydroxyacyl ACP. Then it will be converted to trans-enoyl ACP. And finally, it will be converted to butyryl ACP with the thioesterase enzyme. This unit will be released. And every time 2 fatty acid, 2 carbon are added, and ultimately it will form palmitic acid. We can compare with the beta oxidation. What happened in beta oxidation? Acetyl CoA fatty acid, first it was oxidized to trans-enoyl-CoA, then hydration occurred and it will form hydroxyacyl-CoA, then it will be converted to ketoacyl-CoA. It is converted to ketoacyl-CoA, and then acyl-CoA with the help of thiolase plus acetyl-CoA. This acyl-CoA is 2 carbon less. So same in case of fatty acid. In case of fatty acid, first is 2 acetyl-CoA plus malonyl-CoA. They will form the ketoacyl ACP. Then it will be reduced. Here hydroxyacyl-CoA was converted to ketoacyl-CoA. So here ketoacyl-CoA will be converted to hydroxyacyl ACP. Then in this procedure, trans-enoyl CoA was converted to hydroxyacyl CoA. Here, hydroxyacyl CoA will be converted to trans-enoyl CoA. So in fatty acid synthesis, hydroxy will be converted to trans-enoyl ACP. And here there is a dehydration occur. And this trans-enoyl converted to butyryl ACP with the help of, again, it is reduced, and ultimately butyryl is, with the help of thioesterase, it will be removed. So this is how about the fatty acid synthesis of 16 carbon. Further elongation taking place in the endoplasmic reticulum, and with the help of NADPH, very long chain fatty acid are synthesized. Coming to the unsaturated fatty acid, with the oleic acid can be synthesized by the desaturated enzyme. It will add the fatty acid 2 carbon up to the delta 9. So linoleic and linolenic, they are essential fatty acid. It has to be supplied in the diet. They are not synthesized by our body. While arachidonic acid is semi-essential, it can be synthesized from the linoleic or linolenic fatty acid. Coming to the regulation, the fatty acid, as you know, acetyl CoA carboxylase is a key enzyme of this pathway. Allosteric regulation, citrate and ATP. The more citrate is there, so as we know, in TCA cycle, acetyl CoA plus oxaloacetate, it was converted to citrate. So when more citrate is there, it will channel this acetyl CoA to the fatty acid synthesis. In more ATP, so cell will know it don't require it further energy, so it will channel to the fatty acid synthesis. So citrate and ATP, they will inhibit and they will favor the fatty acid synthesis. Then in regulation, hormonal regulation, so insulin, as we know, it has a phosphatase activity. It will cause the dephosphorylation of the enzyme, and dephosphorylation form of acetyl CoA carboxylase is active. So it will stimulate the fatty acid synthesis. While glucagon, it will cause the phosphorylation. A phosphorylate form of acetyl CoA carboxylase is inactive. So it will inhibit fatty acid synthesis. So glucagon will inhibit fatty acid synthesis. This is about the fatty acid synthesis. If you like my video, please hit the like button. Thank you for the watching. Please share and subscribe to my channel. Thank you.